De Vries from the Allen Brain Institute will be speaking about activity in the visual cortex in the awake mouse. Welcome.
Great. Thank you. Thanks for inviting me to speak. So as we know, the brain uses a wide range of complex and different types of sensory information in order to guide perception and behavior. And how exactly this works is still a pretty open question in the field. And it's a question that we're interested in here at the Allen Institute. So how does the cortex represent sensory information? And how is those representations, how are they transformed by the computations that are carried out by the cortex? So I'm going to tell you about some of our efforts to look into this question, and specifically about a new data product that we're releasing, the Allen Brain Observatory, and this is going to be publicly announced in a couple weeks. The Brain Observatory is a large survey of physiological activity in the mouse visual cortex in the awake mouse. And using high-throughput calcium imaging, we are able to capture the responses of cells throughout the mouse's visual cortex to a wide range of different visual stimuli. And we've designed this to allow us to make comparisons of these responses across different visual areas in the cortex. So here I'm showing you the surface of the cortex with the different visual areas, the primary visual area and the higher visual areas surrounding it outlined. We also want to make comparisons across different cells in different cortical layers, and that are identified by different genetic cell types. So looking at different cortical layers and crete-lines, and using a wide battery of visual stimuli that we can use to really characterize the cell's visual response properties. So we have a number of stimuli, including synthetic stimuli, gratings and noise, as well as more naturalistic stimuli as well. So as I mentioned, we accumulate this data using two-photon calcium imaging. The primary data collection is done when the mouse is awake on a running disk under a two-photon microscope. The mouse is free to run. It's not compelled to do so. The experiment doesn't require it. And so we present different visual stimuli on this monitor in front of the mouse. And while we do that, we then can image the activity of the cells that you can see here in this movie. So we use a transgenic approach. So like I said, we want to look at activity in different populations of cells identified by different crete-lines. So we use crete-lines to drive the expression of GCAMP6 fast. And so this is a calcium indicator. Whenever the cells are active, whenever they're responding to things, you see that they light up. And you can see in this movie, in a field of view, which is 400 microns across, we've got a large number of different cells, and they respond at different times to the features of the stimulus. At the same time that we are imaging the activity, we collect two other pieces of data. One, we have a camera that's focused on the mouse's eye to track the eye position on the monitor as well as the pupil diameter. And we have another camera that captures the mouse's posture and behavior, so we can see if the mouse is running or whisking. And we also then record the speed of the mouse's running based on the wheel rotations. So I mentioned we want to use a wide range of different stimuli to look at the mouse's responses. This includes, I need to find another place to stand, drifting gratings and static gratings. So drifting gratings will move in 8 different directions and at 5 different temporal frequencies, so we can look at the motion preferences of these cells. The static gratings are flashed very briefly, again, 6 different orientations, and now 5 spatial frequencies that let us look at the spatial acuity. We use a sparse noise stimulus to map the spatial receptive field, so where in space a given cell is responding to, and whether it's responding to increases in light or decreases in light, and what the structure of that receptive field is. And then we use natural stimuli, natural scenes, a collection of about 120 different scenes that we've chosen from a number of data sets, and natural movie clips. We have 3 different natural movie clips. As you can see, this is a really large stimulus set that we have. And so in order to collect all of this data, we need to break the stimulus up over multiple sessions. So we broke it into 3 different sessions that each last about an hour. And so this adds a constraint to our experiment in that we need to go back and image from the same cells reliably from one day to the next, accumulating 3 sessions worth of data for each field of view that we have. So how do we actually carry out these experiments? So to do this, we have to use a very standardized method so that we can find our cells, we can go back to them day after day, that we know the visual areas that we're imaging from. We need to do this in a high throughput manner, and so everything has been really highly standardized, and the people who are carrying out these experiments are highly trained in doing them. So it starts with the surgery where we create a window. We remove a small piece of the skull and replace it with a piece of glass. When we also put on a head frame. And this head frame is designed so that every time we put the mouse in under the microscope, it's positioned exactly the same place, oriented the same way with the monitor, regardless of which microscope it's on or which system we're using. This, again, creates a window that we can image through. Each stage has many steps of quality control to make sure that the procedures to date and the data that's being created meets certain criteria so that we can include the data. So we want to make sure that the orientation of the head frame is consistent for each mouse. We want to make sure that the health of the brain and the tissue is all good. After the mouse recovers from surgery, it goes to intrinsic signal imaging. The purpose here, I'm not going to go into great detail, but the purpose is to create a map of where these different visual areas are within our cranial window. So, and to define the retinotopy of these different areas. And so the end result here is a map that shows the different visual areas that then get segmented. And this allows the downstream users to be able to go and target specific visual areas for data collection. And it also defines the targeted location based on retinotopy. We want to image cells that are pointed in the same part of the visual field from each experiment. Following this stage, the mice go through a week of habituation so that they get acclimated to being on the running disk, having different visual stimuli presented to them, so that when we go to collect data, that they're in a relaxed and comfortable state. And then they then transition to the in vivo two-photon imaging. Again, as I described before, the mouse is on a running disk in front of a monitor that presents these stimuli, and we can image the activity of hundreds of cells. And we create, as I showed before, we have our movie of the calcium activity, the movie of the eye position, as well as the movie of the mouse's posture. And we repeat this over many sessions for each field of view. And we actually will image from multiple fields of view for individual mouse in order to maximize the data that we can accumulate. We follow this with another stage of intrinsic imaging to confirm our visual areas. And then at the end, after the mouse is euthanized, it's sent to serial two-photon imaging, which allows us to reconstruct the brain in 3 dimensions so that we can map the cells that we've imaged into the common coordinate framework, which is something we use for most of our data sets that allow us to relate their position to each other. This, of course, is the pipeline for the data production. And there's a lot of controls in place to make sure that this data is good, especially for the two-photon imaging. We monitor hundreds of parameters for each session about image bleaching or whether there's the amount of motion in the movie, that we really scrutinize them to make sure that the data is good. Following the data production, though, there's a lot more that has to happen in terms of processing this data and managing the data. So as I mentioned, we create these maps of the visual areas. These areas need to get segmented. The targeted locations need to be computed from the retinotopy. Once we collect the calcium data, there's a lot of processing here in terms of motion correction, segmentation of the individual cells, registration of these identified cells across the 3 sessions, the fluorescence extraction and neuropil subtraction. This then gets aligned with the temporal information of the stimulus that allows us to calculate visual response properties. And all of this then gets packaged into the neurodata without border file format, which allows us to kind of share this data more easily. Similarly, there's a lot of processing with the serial two-photon imaging done for the brain reconstruction to do the alignment. And so just as there's a pipeline of data production, there's also a pipeline of data processing with a large number of different modules carrying these things out, again, in a very standardized way from each experiment, trying to minimize the human involvement in these things. And so this is just a diagram of some of these modules and the data that's getting passed from one module to the next in this pipeline. So the result for our first release of the Allen Brain Observatory is that we've got a data set that includes over 18,000 neurons that are spread across 4 different visual areas. So data is collected in primary visual cortex, PM, LM, and AL. Each circle here represents a single experiment, and the colors then correspond to the 4 different Cree lines that we've used so far for this product. And then finally, you can see how these experiments have been distributed across imaging depths. So we image at different imaging depths, hopefully trying to get different layers from the cortex where the cells are being expressed. So the table over here can show you the distribution of these neurons and the number of experiments for each visual area and each Cree line to get a sense of the breadth of this data set. So now, for each of these 18,000 cells, we've computed their responses to the battery of stimuli that we've presented. And so for each cell, you can click on the web page and see these responses to the natural scenes, the drifting gratings, the static gratings, the spatial receptive fields here, responses to the natural movies, as well as the speed tuning of the cell. So here we're just looking at how the activity of the cell relates to the mouse's running speed without regard for the actual visual stimulus. So I imagine that most of these figures don't really look very familiar to you. We actually have changed some of our visualizations to try and capture as much information about these responses as succinctly as possible. To kind of explain it, I'm going to walk through our representation of the response for drifting gratings. So I mentioned the drifting gratings stimulus. It moves in 8 different directions and at 5 different temporal frequencies. So that's 40 different stimulus conditions. And each condition has 15 trials that are presented. So here I've taken one cell, and I've taken the 15 trials of a given stimulus condition, so a direction of 0 and a temporal frequency of 1. And you see the change. I plotted the change in fluorescence for each trial. And you see some trials have larger responses than others. What we've done is we've represented these responses each with a single dot, whose color corresponds to the strength of the response during that trial. These 15 dots we rearrange into kind of a radial circle where the strongest response is in the center, and then the weaker responses kind of radiate out from there. And this then gets placed onto what we call the star plot. So on this plot, what we have is each arm represents a different grating direction, and each ring represents a different grating temporal frequency. And so at the intersections, we can place the dots of the 15 trials, and you can clearly and quickly see the orientation tuning of the cell. So this cell prefers gratings that move along this axis with a slight preference for the motion to the right. It's actually probably slightly off axis because of the response along this axis here. And its response is at lower temporal frequencies rather than higher temporal frequencies. But in addition to the tuning information, by capturing the response for each individual trial, you can also start to get a sense of the trial-to-trial variability, which is something I think really interesting about what we see in the mouse visual cortex, is how much variability we see from one trial to the next, particularly in the awake mouse. So that's kind of the philosophy behind most of these representations, where we really want to be able to capture the responses to kind of all of the different stimulus conditions, as well as their trial-to-trial variability as succinctly as possible. So just to kind of explore some of these cells, we can look at this cell in particular. On the website, what's really lovely about these representations is that you can hover your mouse over a part of this plot, and it'll show you the stimulus that corresponds to that. So when you look at the responses to the natural scenes, so this is what we call a corona plot. Each ray represents the response to a particular image, and again, each dot is a single trial of that image. And so you can find one of these. So this cell has a very kind of selective response to just 2 images, and you can look at what's this image, and you see that it's this image of a butterfly, one of the images in our data set. And you can, by interacting with the data, you can get a sense of what its preferences are. So this is an example of a cell that has strong orientation tuning and direction tuning, where it really likes motion that moves to the left. Its response to static gratings is consistent, that it has a strong preference for vertically oriented bars. And one interesting thing in this cell that we see is that it has a really reliable response to the natural movies. So again, this representation has, the movie is a 30-second clip. And we've represented the change in fluorescence where the movie clip begins up here at 12 o'clock and rotates clockwise. Each ring represents a single trial, and the outer blue ring is the mean across the 10 trials. And so by seeing where we see these bands of activity is where trial after trial we see a reliable response. because we have 3 different sessions for our experiments, that we do repeat this. One of the movie clips that we have gets repeated across all 3 sessions, so we can compare the activity of this cell across the different imaging sessions we have. And this cell has a very robust and reliable response to this clip in the same place. And so we can, it's a little bit intriguing. We can look in at the movie and see what's going on. And so I've trimmed this movie just to start kind of just before the first big burst of activity and run until the other hash mark over here. And what you see is consistent with its grating tuning is that this cell is being driven by leftward motion in the scene. So as it starts, one of the first things that happens is this person moves across the field of view, and as he runs away, this wall is moving leftwards with these kind of inlaid posts coming with it, which is what I assume, I'm pretty sure is driving these little bursts of activity in that cluster. But after that, in the rest of the scene, there's no more of this strong leftward motion that's going with it. Here's another example of a cell. In this case, the cell has strong orientation tuning for horizontally oriented bars, as you see both for its responses to drifting gratings, where, again, it responds to horizontally oriented bars, but that are moving in either direction. Again, with the static gratings, it likes the horizontally oriented bars, and at a particular spatial frequency, a fairly high spatial frequency. And when we looked at its responses to the natural scenes and looked at what are these scenes up here that it has a particularly strong response to, it turns out it likes these horizontally arranged pencils. This shouldn't really be shocking, right? If it likes gratings, horizontal gratings, and we've got a natural image that is as close as you can get to a grating stimulus as you can in the real world, it makes sense that these two would be correlated. This is it's cute, it's a nice anecdote, but I was intrigued by this, and so I went through our data set of 18,000 cells to identify the cells. So we have both the horizontal pencils, but we also have a vertical pencil. So I identified all of the cells for which this was their preferred image from the natural image data set. And then I looked at those cells and looked at what their orientation preference was for the static gratings. And unsurprisingly, what we see is that for the vertical pencils, the cells are tuned for vertically oriented gratings, and for the cells that have their preferred image of the horizontal pencils preferred the horizontally oriented gratings. Now, again, this is cute, but I think one of the things that this underscores is that there aren't that many cells in our data set that have these 2 images as their preferred image, right? It's less than 100 each out of 18,000 neurons. So in a given experiment, there might be 1 cell, maybe 2 if you're lucky, that have these preferences. And so if you wanted to make this comparison in a single experiment, you're left with anecdotes. But by looking across this large volume of data, we can actually start to confirm that the visual system, in one way works the way we expect it to. So far I've been showing you some comparisons of individual cells looking across the different stimuli and trying to look at their responses and whether they predict from one stimulus to the next. But I want to consider some of the other types of comparisons that we'd like to be able to make in this data set. So one question is whether we see differences across the different visual areas that we've been imaging in. And so what I've done as a preliminary step is simply to look in each visual area at how many cells have a significant response to a given stimulus. And I've done this for the 2 grating stimuli and the natural scene stimuli. So this is just a statistical test whether they have a significant response to one of the many stimulus conditions for each stimulus. And what we see is that a large fraction of the cells do respond to these stimuli, but when we get into the PM and the AL, there's actually a slightly smaller percent of cells that actually respond to these stimuli. And what is intriguing about this is that these 2 areas are thought to be part of the dorsal pathway of the visual stream. Right? So there's a ventral pathway, which LM is a part of, and PM and AL are thought to be in the dorsal pathway. The ventral pathway is thought to be involved in scene processing, object recognition, spatial acuity, while the dorsal pathway is thought to be more involved in visual motor, navigation, and these types of processes. So it's intriguing that there's fewer visually responsive cells in these dorsal pathways. Looking at this in a slightly different way, what I've done here is look at not just whether they're visually responsive, but whether they show significant modulation by the mouse's running speed. So here, the yellow wedge is cells that have a significant response only for the visual stimulus. The blue wedge is the cells that don't have a significant response to the visual stimulus, but are significantly tuned to the mouse's running speed. And the green ones are the ones that have both, that they're both visually driven and tuned for the mouse's running speed. And again, we see a large number of cells in the dorsal areas that show the speed tuning for the mouse's running speed without being visually driven, which again could be consistent with a role in navigation or spatial processing. Finally, if we want to make comparisons across the different cortical layers, and we've begun looking at various metrics of the visual responses and comparing these across kind of all of these different parameters. And one thing that kind of popped out early on was a difference by depth in the direction selectivity index. So this is a metric as to how direction selective a cell is. A high value means that it strongly prefers motion in one direction and not in the opposite, whereas a low value means that it would respond both equally to motion in one direction and its opposite direction. And what we see is that in these intermediate depths at 275, 350 microns, we see a higher direction selectivity index than we do at 175 or 375. We believe that these depths are close to layer 4. And so it's a bit surprising to see a higher selectivity in layer 4 than in layer 2, 3, or layer 5. This is consistent when we also break it down by the different Cree lines. So both RORB and SCNN1A are layer 4 specific Cree lines, and they both show kind of elevated direction selectivities compared to, say, RBP4, which is exclusively in layer 5. So as you can see, this is a really large data set. We've barely just scratched the surface in trying to get a grasp of what the data is telling us about sensory representation in the visual cortex. And so there's going to be a lot of analysis that can be done with this and a lot of interesting questions to be further explored. I want to just thank everybody at the Institute who's contributed to this. This is over 100 people who've been involved in collecting the data, processing the data, analyzing the data, telling us what to do, and keeping everything moving smoothly. And also want to thank the Institute as well as our founder, Paul Allen, for his vision and support. Thanks.
Well, thank you, Dr. de Vries, for that really technical tour de force and amazing amount of work that you've done and your colleagues have done. We have time for a few questions. There's one in the back there. Get a mic up there.
Thanks for a great talk, and I think this resource is one that's going to be really transformative in the field, so it's really exciting to hear about. I had a couple of technical questions. First of all, I was wondering why you did the wide field mapping with intrinsic imaging instead of the wide field GCAM, since these are GCAM transgenic mice already, I think. And then secondly, I was wondering if you could talk a little bit about the image processing. I'm specifically wondering about the segmentation and what kind of automated or non-automated algorithms you used for that.
Yeah, so we use intrinsic signal imaging rather than kind of evoked, right? There are people who also use evoked activity for this. This was, I think, primarily just a decision based on where we were at the time when we were setting these things up. There were people working on doing it using evoked, and we already kind of had an established method using the intrinsic imaging, and it was working well, and so it was just a decision to go with that at the time. The segmentation, really we should talk a little bit more offline, but it's a method that basically does kind of like a local thresholding to identify the segments, going through kind of small pieces at a time and looking at the morphology. There's certain criteria on shape and size that are used as well, but it's pretty complicated, so we should maybe talk about it afterwards. But
Hi. Oh, hi. It's a great talk. I was just wondering, a simple question. The way I understand it is you're using largely video, so even though you have three-dimensional objects, they're still on a 2D screen. So if you actually had actual three-dimensional objects moving around, that's in what's at the rat's or the mouse's field of view, how that might change the maps, if at all.
I have no idea. I think that, if we were to, it's a great question. I think one of the directions that some of the people in our institute are interested in is object recognition, right? And that's one of the questions is when you see an object and it's at different angles, how is that representation going to change? And that's something that in the ventral system, you see changes in where early, say in V1, you might expect different responses based on like the orientation of that object, whereas in higher in the ventral stream, you expect to see more, the responses will remain consistent for a given object. And so this is a direction that we want to be moving in kind of future projects. But so far, none of our data really speaks to that. But that is kind of an intriguing thought of how those transformations could affect these responses, right?
Yeah, one more question.
Hi, I just want, you just mentioned about the trial-to-trial variability. I'm wondering if you could just expand on that a bit and give us some insight on how much, like the range of it and how much is not accounted for by things like running speed and all these other variables that you measured. Thanks.
Yeah, that's a great question. So the mouse's running speed is perhaps one of the largest factors for the trial-to-trial variability, and just in terms of like the extreme size. So the example I showed you of the 15 trials for that drifting grating, and there were probably 5 or 6 that you could see the kind of the modulation of activity, and the rest were almost at baseline, but part of that was just that scale. And if you were to zoom in on the other ones, you'd actually see that there was a response there, but it's just much smaller. And those trials that really stood out are the trials when the mouse was running compared to the other ones. So it's not that the mouse wasn't responding when it was stationary, but that the modulation by running for that cell was so strong that it really kind of exacerbates those differences. Beyond that, I mean, even in the anesthetized mouse, we do also see a lot of variability compared to what you expect, what you see in cat or primate. So I can't really speak to all of the factors that are contributing to this, but I do think that in the awake state, you've got the running activity, just the kind of whether the mouse is paying attention or not, because it can kind of sometimes get bored and tune out. And all of those are going to be really big factors on that variability. And so, hopefully using the pupil diameter, we can get a sense of the arousal state of the mouse independent of its running speed and see if that could also be influencing these things. But it's something that I think is going to be very rich to explore.
Well, thank you again, Dr. De Vries.